C'est bon, vous pouvez rentrer, hein. Oh, Roman, it's Roman. He was young, no? Younger than now, no? Good picture. And to be
Okay, so hi everyone. So welcome to the Montpellier series seminar in ecology and evolution. So before we start, just a few technical issues, especially for those who attend on Zoom. So just a reminder that you can ask questions via the Q&A tab, and that you can use the chat if you have any technical issues. So the seminar will be recorded and posted on YouTube. And with that, I leave it to Marie to present the speaker.
Oui, merci beaucoup. Merci d'être venus nombreux, c'est cool. C'est vraiment un grand plaisir d'accueillir Cathy aujourd'hui. Voilà, je suis très contente de l'avoir parmi nous. Donc, en quelques mots, Cathy Crockford, elle a passé sa thèse en 2005 au MPI, donc le Max Planck Institute, et à l'université de Leipzig en Allemagne. Et après 2 post-docs qu'elle a fait aux États-Unis et en Écosse, je crois, elle a été recrutée en tant que groupe leader au Max Planck Institute, toujours de Leipzig, où elle a obtenu entre autres une ERC consolidator pour travailler sur les effets maternels sur le développement des jeunes chimpanzés. Alors Cathy, elle est surtout connue pour co-diriger depuis de nombreuses années un projet de long terme en fait sur le terrain pour étudier une communauté de chimpanzés dans la taille forest en Côte d'Ivoire. Voilà, donc Cathy travaille sur de nombreux sujets, dont l'évolution de la socialité, la cognition sociale, la communication, la coopération, etc., etc. Et depuis, en fait, récemment, elle a été employée, elle a obtenu le concours de directeur de recherche du CNRS. Et aujourd'hui, pour notre plus grand bonheur à tous, et elle est aujourd'hui donc chercheuse à l'Institut des sciences cognitives à Lyon. Et voilà. Je crois que c'est à peu près tout. Merci beaucoup, Cathy, d'être là. Et bon talk.
Merci, Marie. C'est très gentil pour être ici. Je vais pas parler en français, pardon, parce que je pense que vous voulez comprendre plus si je parle en anglais. So that's the end of the French, sorry. But it's really a great pleasure to be here and in person and to have an audience. And thank you very much, Marie, and all of you for being here and for inviting me. And also, it's very nice being new in France and new in the CNRS to come and see another institute. So this is all very exciting for me today. I'm going to talk today about oxytocin and cooperative behavior in wild chimpanzees. I think maybe I take my mask off. And whether this gives us any insights into human cooperation. This is work that I do with 2 very long-standing important colleagues of mine, Roman Wittig, who's the director of the Thai Chimpanzee Project in Ivory Coast, and Tobias Deschner, who until recently was the head of the ape endocrinology lab at the Max Planck Institute in Leipzig, and also works in a field site in Gabon, not too far from Marie. And so let me start by posing a question to you. What is it that makes you switch from being competitive to being cooperative? Right? So, you are interacting with your family, your friends, your colleagues, and strangers. And sometimes you will, for example, be aggressive, and sometimes you will be helpful. Right? And this is the same for many animals. For example, these chimpanzees with this piece of meat, they can choose to fight over it or to share it. So what is it about these interactions that determines how we engage? So luckily, the neurophysiology, the neuroendocrinology that impacts on behavior is quite conserved across mammals. And so if we want to find out something about our own behavior and what determines it, it's quite interesting to then look at other species, that maybe we can address these questions a bit more easily in. So, oh, it's not working. Voila. Sorry. Okay, let me start by thanking my team. So this is mine and Roman Wittig's lab, and the work I talk about today will be from Paddy Kaczynski, Loran Samuni, and Anna Price. And I want to thank also our fantastic team of field assistants in the Ivory Coast, and also other key collaborators like Kevin Langegraber and Linda Vigilant, who've done the genetics for us, the hormones with Tony Ziegler, as well as with Tobias Deschner, and for cooperation across field sites with Klaus Rubula. So the starting point here is social bonds. Okay, so social bonds we can define relatively easily as high rates of affiliation within dyads. And we find these most across mammals in pair bonds, but also we find them in kin bonds, for example, in the cooperative breeding wood hoopoes, and also in matrilineal primates, for example, these chacma baboons with a baby, a mother, and the grandmother. But these bonds between the adult females can be maintained throughout their adult lives. Much less frequently, across the animal kingdom, are non-kin bonds. Okay, we have these in humans, and they have been exploited by the military for several thousands of years, at least starting with the Spartans, and here we see some army guys. But they're not very common in other species. We do see them in chimpanzees. So they have these bonds for years and years with individuals where they have very low relatedness. Okay, so how, what shapes these bonds? How do they exist? Another important thing about social bonds that we have in the last 20 years found out a lot about in primatology, and this work has spread now to other, throughout animal behavior, is whether social bonds have fitness consequences. And we find that for an individual in a social group who has social bonds compared to one who does not, they have more offspring, they have increased longevity, and they have reduced stress and disease. And there is a correlation here also with cooperation. So that those who have social bonds tend to cooperate, for example, in dyadic coalitions, and this cooperation also leads to more offspring. So what it seems, a hypothesis that we now have, is that those who share bonds are more likely to cooperate, and in fact, that it might be the bonds, this ability to form bonds, that facilitates the cooperation. So if you're a species that benefits from cooperation, you're quite likely then to also have the capacity to build social bonds. How does this look like in humans? Well, there's this very famous study from Holt-Lundstedt, a meta-analysis with 300,000 participants. And she looked at the social relationships and the mortality risk related to that. And she compared this risk with other known mortality risks. Okay, so we have the effect sizes along the top, and obesity has, small mortality risk, alcohol drinking more so, smoking even more. But most of all, the factors that had the biggest influence on mortality risk were social support and social integration. So the message here is clear. It's fine to eat, it's fine to smoke and drink as long as you do it with your friends. So the problem is that we don't know what is causing this relationship. Is it more about the emotional support that you share with somebody? Or is it the cooperative exchange? And this kind of detail is difficult to address in humans because the data relies on self-report. It's ethically difficult to do direct observations on people. So the self-report questionnaires is what feeds a lot of these studies. And then the kind of variables that they're looking at are proxies. So for example, whether you go to church or not, whether you are divorced or not, the kind of questions on this questionnaires that determine your ability to form relationships or be socially integrated, which is a problem in my view. But measuring relationships and social interactions directly is our bread and butter in primatology and animal behavior. This is what we do. So we have a really nice opportunity to try and get a closer look at these kind of questions. So in primatology, one of our standard sampling tools is focal animal sampling, where we get to know the rates of grooming and who individuals are grooming with across years and now sometimes across decades, because the field sites have been running for a long time. And we typically get distributions of grooming that look like this, with a lot of left-hand skew, so that there are, in each social group, a lot of dyads that never or rarely groom each other. And then there are very few dyads that groom a lot, okay? And in these individuals, the grooming tends to be mutual and enduring, so that these dyads can last over years and years. And therefore, by definition, in these dyads, the grooming is predictable. Animal A knows when it approaches animal B that it's more likely to receive grooming from that individual than from any other individual in its troop. Okay? And these ones we call bond partners. And if they are not kin, we call them friends. Okay? And these friends, bond partners, are likely to come to the other's defense in an aggression, right? So there is more cooperation and predictable cooperation here. So in chimpanzees, this can result in injuries too, like this. So what is it that makes one individual come and risk injury to save another one, even though they're not related? This cannot be explained by indirect fitness benefits, right? It can only work through direct fitness benefits. So one thing that we wonder is, is this held together through some kind of emotional connection? Emotion is hard to measure, but we can look at underlying hormones. And a relevant hormone here would be oxytocin. And we ask whether it facilitates these adult relationships, these adult non-kin friendships, and supports cooperation. So oxytocin is well established now to be critical for parturition and lactation across mammals, and also for establishing the mother-offspring bond. So now what about when lactation is finished? How do these bonds continue? For example, like I said earlier, in these matrilineal primates, who the mother and grandmother will stay friends for life. There's no lactation now to keep this together. And in bonobos, the mother and her adult male son, and then in the chimpanzees, in these non-kin bonds. So one idea from Curley and Cavern is that the mechanisms, the neuroendocrinology that facilitates the mother-offspring bonds is co-opted to be used in these adult relationships. So we wanted to explore this idea and see if we could find some support for it with empirical data. So the first thing was to think about behaviors that are used from a mother to her offspring, and those same behaviors that are used in adults. So in chimpanzees, this would be grooming, obviously, but also food sharing and defense of others. So we took these three behaviors as nurturing in the mother offspring, and then described as cooperative when it's about between adults, because it needs to be reciprocal. And we asked if these behaviors are associated with oxytocin release. Now we're studying wild primates, so we want a non-invasive approach. So we look at endogenous oxytocin, And for this to work, here is the hypothalamus. And the yellow neurons are the ones that produce oxytocin and distribute them to the rest of the brain, which is what changes our behavior. Okay? These green ones are the neurons that distribute oxytocin into the posterior pituitary, where it goes into the blood and becomes peripheral. So in order to measure peripheral oxytocin and for it to have relevance to behavior, the idea that there needs to be synchronous coordinated release of these two types of neurons, so that we know then that what we measure in the periphery, in the blood, the saliva, the urine, is saying something about what's happening in the brain. And there are a number of studies that do support that there is coordinated release, and a study that some colleagues of mine did that a lot of students signed up for to be given MDMA. And they did find it, because this releases oxytocin in the brain, and they did find that there's high oxytocin in the blood and the urine concurrently. So this is a nice validation study. And also, urinary oxytocin has social correlates in primates, for example, to do with their pair bond strength and mother support, which made us decide to go for urine as our substrate. It's a nice substrate because once oxytocin arrives in the bladder, it is stable in the urine, and then you can get an accumulative signal, so you can get quite a nice, strong, robust signal compared with saliva or blood. And we wanted to assess the oxytocin related to single social interactions. Okay, we call this event sampling. How did we do this? Non-invasively and in the field. Well, first we found our chimpanzee site. We started in the Bodongo forest in West Africa, in East Africa, with one subspecies, and then we switched to West African chimpanzees, another subspecies. The genetics we established the genetics for these different populations. And then this event sampling approach relies on the clearance time it takes from oxytocin to arrive in the urine. Okay, and this is about 15 to 60 minutes. So if we have a grooming event from 8 to 8:45 in the morning, then we need to collect the first urination that happens within that clearance time window. Any other urination is too late, and we abandon this event. It's also better if there is no social interaction in the preceding hour or in the hour afterwards, so that we can really attribute the oxytocin level to this single social event. So once we have observed our target behavior, then we keep our eye on the butt of that chimpanzee until it pees, and we collect the pee either in a plastic bag if it's up in the tree, very technical, or we pipette it off leaves. And then at the end of the working day, we place it in liquid nitrogen. And when the tank is full, we ship it on dry ice back to, in the early days, to Wisconsin, and then more recently to Leipzig, where the samples are extracted and then subjected to immunoassay, and then we control for water content as well. So the first, we wanted to look at this nurturing idea, right? This co-opted mechanisms idea. So we looked at the first nurturing behavior between mothers and offspring. It also occurs in adults, which is grooming. So we looked at grooming between non-friends, and we looked at grooming between friends. Okay, so those that groom at very low rates and those that groom at high rates. And what we found is we compared these with a 1-hour rest sample. Okay, is our kind of baseline. And what we found is that at least 10 minutes of grooming between non-bond partners showed similar urinary oxytocin levels as the 1-hour resting. However, grooming periods with bond partners showed a significant increase in urinary oxytocin, or in higher levels. This was the same whether the bond partner was kin or non-kin. And also, it was the same whether they were giving grooming or receiving grooming, okay, which I find particularly interesting. So it seems here that the oxytocin release is not to do with the act of grooming itself, but to do with the relationship that the dyad shares around the grooming event. So it seems that oxytocin is somehow related to supporting these social bonds. So then we looked at this in the second nurturing behavior, food sharing. So in mother offspring, this starts off with lactation and then it moves on to solids. And in adults, they're sharing things like meat and honey, typically high energy foods that need some kind of extractive work. So here we have it. I'll show you a video. I hope you'll be able to see it. It's not that you can see with the quality. But there's two adult males here. One has honey. He has honey in both hands that he's found. He had to dig it out and he had to get stung by bees, so it took some effort. And in chimpanzees, they can be very aggressive to each other. They can kill each other. But those who share bonds, affiliation is predictable. Food sharing is predictable. So what you'll see here is a very calm interaction of food sharing, where one guy, the alpha, actually asks the other guy for food. He puts out his hand and the other one releases it, and then they sit and eat together. So there he is eating the honey. The other one comes around, you'll see him put out his hand, and the other releases a big piece of honey, and then they sit very calmly and eat together. This little orphan here at the front does not get in. So yeah. So when we look at the oxytocin levels, this is the grooming data I just showed you. If we add the food sharing data, you can see that the oxytocin levels are significantly higher for the food sharing events than they were for the grooming events. Okay, so even though there isn't necessarily even any physical contact and it's a quick exchange, it somehow has this big impact on oxytocin release, apparently. So then we repeated this to see if we get the same results on a different population of chimpanzees in West Africa with a different hormone lab, and we found the same result. This is Laurence Louise, part of her PhD work. So it seems that this result is replicable and robust. So then we looked at our third, another behavior, and nurturing of defense of others, right? So, and we looked at also a second group. So chimpanzees are also unusual in that they don't only have these dyadic coalitions, which a lot of animals have, but they also work together as a group, right? So they will go on territorial patrols, and they'll hunt monkeys, and they do this with very coordinated behavior. So we wanted to know how these group level cooperative events affect oxytocin, or what the correlation is there. So when they're hunting monkeys, the oxytocin levels were similar to when they're food sharing. But when they are defending their territory, when they're cooperating to defend their territory, the oxytocin levels were significantly higher than the food sharing and hunting. This is a logarithmic scale. So, it seems that these cooperative behaviors are related to high oxytocin, but some cooperative behaviors produce even higher oxytocin levels. Okay. And let me show you a video of what that behavior looks like, because it's quite hard to describe. So what you'll see is chimpanzees meeting in the core of their territory. Unfortunately, I won't put the sound on, but they are loud at the beginning. There's a lot of greeting behavior, a lot of very intense affiliation. So testicle holding, you'll see, for example, And then they head off to the periphery of their territory. They become extremely quiet. Their behavior becomes quite coordinated. So they stop at the same times, they walk at the same times. And then in the final moment of ambush, one of them has to start, right, and run forward. Now, if the others don't follow, that guy will be killed. So he's got to know that the others have his back, right? So it's a really dangerous moment. So I tried this now. So you can hear very loud and some testicle holding. I can't see very well from here, but you might see it. Yeah, and now I'll put the sound on again because now they're reaching the core of that the periphery, and it's very quiet now. So very quiet, and they're looking for secondary cues of activity, so sniffing the ground. And if they find nests, old nests, they will climb into the nest to smell. And now here they're getting closer, the behavior is more coordinated, they're stopping frequently to listen. Then, you know, again, they walk forward together. And now in a second comes the moment of ambush. Okay, this one at the front, he's going to lead, and he's, you know, if the others don't follow him, he's dead. There he goes. He runs forward, and it's very noisy. Okay, you get the idea. So then there is this battle that ensues. So this kind of events are somehow related to these very high oxytocin levels. So this led us to ask the question, what is it exactly? You know, this is a correlational approach. We can't really look at causality, right? So what is it that's really triggering this oxytocin release in these cooperative contexts? Is it really something about the cooperative behavior itself? So the coordination, something like trust or shared intent, and that then, you know, the function here would be to limit defection, right, at these critical moments? Or is it just simply exposure to stressors, which has been demonstrated, for example, in rodents? So intergroup encounters are obviously very stressful events, and so food sharing can also be stressful events. So could this be what's triggering the oxytocin? Because there does seem to be a feedback loop between oxytocin, which can down-regulate the HPA axis, lowering the production of cortisol, for example. So we wanted to try and tease these two apart. To do that, first of all, we needed to establish what's really a stressor for chimpanzees, what is associated with the release of cortisol. So we looked at that, and we used an event sampling approach again, and took the clearance window of cortisol and glucocorticoids into account in our approach. And we looked at aggression, because aggression can be deadly, as I said, even within groups it can be deadly, but you know, most aggression isn't. So we just looked at, you know, regular aggression to see if this is also acting as a stressor. So we had our 1 hour resting samples, and an aggressive event showed an increase in urinary glucocorticoids. And similarly with territorial activity, so intergroup encounters and patrolling. When there's no territorial activity, this is the urinary glucocorticoids, anticipation of activity, of territorial activity, and then high GCs for actual territorial activity. So both of these aggressive events are acting as stresses. So then we went back to our question about whether oxytocin is released purely as a result of the stressor. Okay, so this was part of Anna Price's PhD work. And as a first step, she tried to replicate and could replicate our affiliation and grooming results from some years earlier. And then she looked at affiliation post-conflict. Okay, so you've got the aggression, but then you have an affiliation afterwards. And she found whether there was reconciliation or third-party consolation, there was higher oxytocin levels than baseline. So what about if there's just an aggression alone without any follow-up affiliation and no increase, no high oxytocin in these situations? So it seems that it's not, our results are not driven by exposure to the stressor itself, but is something more to do with the cooperative act itself. So then a last study to show you is what happens if you cooperate often, right? So we have this situation. We have several groups of chimpanzees in the forest, and one of them is extremely cooperative compared to the other one. So they engage in much higher rates in these food sharing events, hunting events, and also patrolling events. So we wanted to compare the reactivity in this group that has this high cooperation with a group that has lower rates of cooperation. So yeah, here you can see the aggression samples, the affiliation samples, the post-conflict samples, and the group level cooperation, so the patrolling examples. And then with the other group, these are all a bit less in terms of oxytocin levels. So this is where we are at with this. We don't know what's driving this. Is it that there is some kind of genetic predisposition in one group to have higher oxytocin reactivity, and this is driving the higher rates of cooperation? Or is it the other way around, that higher rates of cooperation are making the release of each cooperative event associated with more oxytocin? We don't know which way around it is yet. So that's where we leave the story for now. And just to summarize that, oxytocin does seem to be important in cooperation. Social bonds and friendship seem to be associated with oxytocin release. Nurturing behaviors give high oxytocin too. So nurturing behaviors like the grooming, the food sharing, and the defense, and then in adults, these cooperative behaviors. So it supports the co-opted mechanisms hypothesis. And that it seems to be something about the cooperative act itself and not the stressor that seems to be giving high oxytocin release. So what might be the implications of this for humans? So obviously we still have a lot of work to do to find that out. But very briefly, maybe one can say that if you want to have a long, healthy life, then maybe balance your competitiveness with enjoying giving and receiving with your friends. Thank you for your attention.
So all we read is a question.
So I was just wondering if you looked at if there's an effect of hierarchy.
At the hierarchy?
Yeah, because if you're younger in a group, you're always trying to fit in, especially for a male as well. So it's very, I could say, chimpanzee ritual to groom your elders.
Yes.
In order to have access, a future prospect for a female in the group. because they're social animals?
Yeah, no, that's a lovely question. Shall I repeat it? Or yeah. So the question was, chimpanzees are very hierarchical. So how much have we really examined hierarchy in this, in the context of oxytocin? So we have always controlled for dominance differences, right, in all of these dyads, but we haven't specifically looked at the effects of dominance directly, right? But one thing I can say, you mentioned that chimps tend to groove up the hierarchy, right? And so this is why we're quite careful with our grooming dyadic measures, that being at high rates of grooming is not really enough. They need to be mutual. So if it's really just about a young guy trying to get attention from an older guy, they wouldn't fall into the social bond category because the grooming would be more or less unidirectional. Yeah. So, yeah.
Sorry.
Good.
Was there an effect with gender? For example, between male and female?
So it's interesting because, so the question was, is there an effect of gender? And this is interesting because chimpanzees are again slightly unusual amongst mammals in that they're male bonded. So there's many more female bonded, or at least in primates, many more female bonded species. And chimpanzees are male bonded. So when we started this study, a lot of people thought there was no point even at looking at oxytocin levels in females or female grooming. And having observed chimpanzees, I was slightly outraged that people would suggest this because, from my observations, it looked like there was also bonds between non-kin females, right? So we included females and the effects were just the same in both species. So actually, we controlled for this. So we haven't directly tested the sex effects. But we plan to do this. But obviously, in females, you need also to control for reproductive state because of lactation. But essentially, you get exactly the same patterns irrespective of the sex combination. So whether it's male-male diet, female-female diet, or male-female diet. Yeah.
Wondering if work has been done experimentally administering oxytocin or oxytocin companion to chimpanzees that imply by reactivity to perform cooperation experiments?
Thank you. Yes, so the question was whether we have done any oxytocin manipulation, so whether we have administered oxytocin in experimental settings to chimpanzees. And the question is that we have, thought about this quite often over, whiskies late at night, how we could possibly do this in a field site. And the bottom line is we realized that actually there wasn't a way we could think to do this that would be ethical, right? Because supposing we administered oxytocin to a subordinate male and he feels, ecstasy, everybody's friendly to me, and he goes up to a dominant male and then he's attacked, right? So in the end, we didn't try it. And I don't think anyone has tried it with chimpanzees. Of course, there are many other studies in other species where, especially a lot, I think, in rhesus macaques, which show all sorts of interesting patterns. I think it's taken quite a long time to work out the best way to do this, how to really make sure that the nebulizer, the oxytocin is reaching into the brain, if it is at all reaching into the brain, what's the correct dose, all of this. So I think it's difficult to do, but I think it's very interesting work. Yeah.
We have a question from Vandana Krishna, who asks, are non-kin social bonds common across the entire group?
So I don't have to repeat that one, do I? No, you said it. Okay. All good?
No, it's just that actually we could turn on her mic and then she arrived. Anyway.
Okay. Yeah, so the question is whether non-kin bonds are common across the groups, right? And actually they're not. About 5, so, you know, in the populations we've looked at so far, whether it's in eastern chimpanzees or western chimpanzees, it's about 5% of all possible dyads. And I think this is a bit less than it is in, for example, baboons, where I think it's more like 8 to 10%, but those are kin-based dyads. So if you know, there's about 5% of kin dyads and about 5% of non-kin dyads in chimpanzees that have these very high bonds. What we want to look at is how many bonds does each individual have? And we don't know exactly yet, but we think it's between 1 and 3 for most individuals. So it's very selective, but the interesting thing that in spite of it being so few dyads, that it seems to have this huge effect on health, longevity, and reproduction. Yes. Yeah, and I really like that result. You know, I expect it would be, sorry, the question was in food sharing, when we look at those who did the sharing and those who received the sharing, do we find a difference in the oxytocin levels? And we actually found that the oxytocin levels were the same. I think that this is extremely interesting because, you know, again, you know, before we started this, people thought that it would only be the receiver who would show some kind of oxytocin change, right? If anybody, right? And the fact that the giver also experiences an oxytocin change is probably something to do with the reward system, right? That keeps this behavior going. Excuse me. So, you know, you need, I'm quite impressed how biology, nature is quite reward driven, behavior is quite reward driven, right? So all the things that are incredibly important to keep a species continuing, like reproduction, are focused around huge reward hits in the brain, right? And so that you find this for something like giving a cooperative act as well, I think is very interesting.
Are there other questions? Actually, me, I have one.
Yeah.
So have you looked at or are you expecting, or perhaps you test that, an increase of oxytocin with the longevity of the friendship of the dyad, of any given dyad?
Yeah, that would be great.
Okay.
And we haven't looked at it yet.
Because it could explain also the effect of the group effect that you have, like variation in cooperation levels and oxytocin levels across those groups. Perhaps that those groups are composed of individuals that are not together for a long time, I mean, new groups.
Yes. Yeah.
Did you look at that?
Well, no, I mean, I can tell you it wasn't that, but we haven't officially looked at it yet.
Okay.
But I think it's really interesting, and certainly it could be a factor, and even, you know, within groups, you know, older dyads could have, you know,
especially because like a grooming event among non-friends has lower oxytocin levels than friends.
When we have a big enough sample size.
Yeah, in 10 years.
In 10 years.
Okay. Yeah.
Do you know how friends become friends first?
So the question was, how do friends become friends in the first place? And we don't know. But Roman, quite, you know, our strongest data is to do with the food sharing data, right? Because here the bond matters less. Right?
What is it?
The bond matters less. So in the grooming, you know, the bond is really important. If you don't have a bond, you don't seem to get this high oxytocin level. But in the food sharing, you can also get higher oxytocin without it being a bond. So it might be that you use the food sharing, you know, whether deliberately or just, you know, through some other process to initiate friendships. Yeah. And we have anecdotal evidence that is quite interesting on that. So that, for example, there's some politicking that goes on in male hierarchies, right? So we had a beta male that was competing very heavily with an alpha male, right? And the alpha male would capture a monkey and very unusually not eat it, but go and take it to the third male and put it at his feet. And they were not particularly friends at that point. And then, you know, a relationship developed, and, you know, it's totally anecdotal, but we were impressed because it was such an unusual behavior.
About this friendship, does it start in childhood or?
Sorry, couldn't you repeat?
This friendship, can it start in childhood or can it also start in childhood?
So the question was, when do the friendships start? You know, do they already start when the chimps are still immature, or is it only in adulthood? And again, you know, data is small on these questions, but I think it's a really fascinating question. It's females that transfer, so this can only apply to males. And I don't know the answer yet, I'm sorry. Yeah. But we will find out. Give us another 10 years. Yeah.
Yes, thank you for your talk. Such great data. I just wonder if you could clarify your hypothesis about the relationship between oxytocin and stress. You said that maybe your results in the condition where they had to cooperate in a stressful way could be explained by stress rather than oxytocin. But my understanding is that adrenaline goes against oxytocin whenever you have a surge of adrenaline. For instance, when you have to give birth and you go to the hospital, there's a lot of light, and you don't know you have a surge of adrenaline, and therefore oxytocin decreases a lot, and you stop labor. So to me, it would have gone the other way. Stress would have explained lower level of oxytocin during the stressful interaction, rather than higher. And it just makes me think that then, how can we use oxytocin to explain cooperative behavior in very stressful contexts? What is the assumption about the relationship?
Yeah, no, that's a great question. I'm just going to summarize it. So the question is, what is really the relationship between oxytocin and the stress axis, right? So, you know, if you're in labor and you're in this anxiety-inducing atmosphere of a hospital, it inhibits actually your progress, right? One would expect then that oxytocin is not really released when there is a stressor, okay? So how does this work? Well, in rodent studies, they have done now over the last 15 years a lot of these kind of tests to try and understand this feedback loop between the two systems, right? And if they do a totally non-social task, like forced swimming, they get concurrent cortisol and oxytocin release. Okay, so this hasn't been shown really very, you know, it's really hard to show in other animals. We don't see this. And it might be that, you know, in our urinary protocol, you have to have really huge increases to be able to detect them, right? So it may be a kind of amplitude thing in terms of the oxytocin level, because one idea from the rodent studies is that if you are exposed to a stressor, it is adaptive to then go and seek social support, right? So that if you don't have access to the social support, there's nothing that can mitigate your stress levels, right? But if you can use this little bit of oxytocin to make you want to go and find social interaction, and you find one, then maybe this is the social interaction then rather than the oxytocin that helps bring down the stress, right? So there's all of these kind of dynamics that it's not really clear yet. And it could be that something similar happens in chimpanzees, but our assay can't detect low levels of oxytocin. So what we can say, at least from our assay, is that the release of oxytocin following aggression is nothing on the, it's not of the same, if it's happening, it's not of the same magnitude as if you actually engage in a cooperative event. Yeah.
Okay, is there a question? Yeah, go ahead.
Another question. I just wanted to know if you looked at the ratio of females in group, and if they are in their breeding, like if they're in their breeding state already, they've bred, or if they're like having children, to see like if there's like a difference based on like females, because
Yeah.
I would assume that if there are non-receptive females in groups, that there could be a bit of more aggression in this group.
Yeah. So the question was about the kind of ratio of nulliparous and parous females, right? And if this is changing the aggression dynamics in the group. I don't know. I mean, I think in chimpanzee societies, most of the females are parous, and then there's very few nulliparous. So I think that's a general kind of structure, and all of our groups would conform to that, right? So that's tricky. But we have a whole new study that I'm really sorry that we haven't presented for you two today, but about mother offspring oxytocin and the interaction with maternal loss and oxytocin, mothering styles and oxytocin. And our very first results suggest that mothers who invest more nutritionally into their offspring, not actually about affiliation, not grooming, but if they invest nutritionally, that they have higher baseline oxytocin levels in mothers. So, it doesn't really answer your question, but that was the only thing I can tell you about parous versus non-parous. Yeah.
Okay. Yep. Other question? Okay. So thank you very much, Cathy, for your talk.
Thank you all.
So now I have many questions for you.